Okay, so this slide here is basically just meant for you guys to use to help you study and familiarize yourself with important terms. So I'm just going to go through these quickly. If you'd like to skip ahead and study them on your own time, you can go ahead and fast forward. But we'll start with cytotoxic T cells. So those are important because they're the only T lymphocytes that directly attack a pathogen or an antigen presenting cell to deliver that lethal hit of chemicals. Helper T cells are important because they're involved in both cellular and humoral immunity, and their job is to release cytokines that draw other lymphocytes or leukocytes into the site wherever they're needed. Memory T cells are responsible for that remember step in both humoral and or memory T cells are responsible for the remember step in cellular immunity. Regulatory T cells are involved in downregulation of T cells. So after we've defeated a pathogen and we don't need this immune response anymore, regulatory T cells are what are going to stop the secretion of cytokines and stop the production of T lymphocytes so that we don't get damage to our tissues. Macrophages are important because they digest antigens and chop them up into little pieces and can act as antigen presenting cells so they can present those epitopes, which are the active part of the antigen, on the surface of their cells to help present them to other cells, like T helper cells or cytotoxic T cells. B cells are different from T cells because they produce plasma cells which make antibodies, and antibodies basically have a different approach. They're involved in humoral immunity. They don't necessarily destroy a pathogen. They can neutralize a pathogen or an antigen, or they can mark that antigen for destruction. But unlike T cells, they don't actually cause any destruction to the cell. Memory B cells are produced from B cells, so the B cell goes into this cloning state, and we get many plasma cells, which we'll talk about in a second, and then we get memory B cells as well. Plasma cells come from B cells that are cloned in response to an antigen. So if you remember, the B cell will take in an antigen, chop it up into little pieces, expose the epitope of the antigen on the surface, on the MHC protein. It will then come into contact, that epitope will come into contact with a T helper cell, which will secrete interleukins, and in response to the interleukins, those B cells are going to start cloning themselves. So you'll get plasma cells from those B cells, and you'll also get memory B cells. So the plasma cells that come from those cloned B cells are going to be responsible for the production of antibodies. Naive lymphocytes are basically B cells and T cells that have passed positive and negative selection. They are immunocompetent at this point, but they're like soldiers that are prepared but haven't yet been to war. So they are immunocompetent, but they haven't come into contact with any pathogens. Antibodies are, again, how we mark antigens or neutralize antigens throughout the body. They come from plasma cells, which come from B cells. And they're part of the humoral immunity. Antigens can be free-floating, like venom, like free-floating molecules, or they can be on the surface of on the surface of cells like bacteria, and they serve as a way that we can recognize our own cells from cells that are foreign. Epitopes are the active part of antigens. Some antigens have more than one epitope. They're just the part of the antigen that actually binds to the antibody and makes it active. Haptens are like half antigens, so they don't stimulate an immune response on their own, but they will bind to other immune cells or bind to other macromolecules on cells and at that point become a complete antigen and basically stimulate an immune response. Complement proteins, I'm not going to get too much into those. We talked about those with innate immunity, but their involvement here is that antibodies basically bind to complement proteins, which then bind to pathogenic or foreign cells, and the complement protein is what actually does the destruction, not the antibody itself. Perforins and granzymes are used in innate immunity, but also by cytotoxic T cells in adaptive immunity. So they are the little proteins that basically perforate a cell. That's why they're called perforins. And then they secrete granzymes into that little perforation, which will destroy enzymes inside of the cell to cause the cell to die. Cytokines are just chemicals that leukocytes use to communicate with each other. Interleukins are one of those cytokines that we talked about, and tumor necrosis factor is just another way that cytotoxic T cells can also kill target cells. All right, this table I'm not going to get too much into. It's just a nice summary of all of the things that we talked about. It's in your book, and I did take some of the stuff out of this table that I didn't necessarily think was important for you guys, and I didn't want you to get too bogged down in, so this is a nice summary if you like tables and stuff like that to study of what we talked about with cellular and humoral immunity. Alright, so now we're going to talk about immune system disorders, and the first of the disorders that we're going to talk about is hypersensitivity. So hypersensitivity is just basically a big, excessive, really harmful immune reaction to different antigens. So we can see hypersensitivity as part of a reaction to transplanted tissues. Sometimes people have these hypersensitivities to their own tissues, which is called autoimmune disease, and we'll talk about that. Or they can have them in reaction to environmental agents or antigens that occur in things like food, mold, dust, pollen, vaccines, venoms, pet dander, plants, and drugs. And there's 4 types that we see here. We're going to talk about each of them individually. So the first is type 1 hypersensitivity, and it is distinguishable from the other types because the response is really rapid. Okay, we get a response within seconds of exposure, and then it usually goes away within about 30 minutes. And so this type of response is what we observe most commonly with your typical allergies. So in this response, allergens, which you see here in this picture in green, are going to bind to antibodies, specifically IgE, on the surface of these basophils. And as a response, that basophil will be stimulated to secrete histamines, which you see here, and other chemicals that have inflammatory effects or affect your blood vessels. Okay, so they'll result in your glands starting to secrete fluids or whatever it is. If it's your lacrimal glands, maybe they'll secrete tears, or your salivary glands, you'll start to salivate. It also results in vasodilation and smooth muscle spasms. So that will make a lot of sense with all of these clinical signs that we see, like edema, mucus hypersecretion. So remember we said glandular secretion, so you get things like congestion, watery eyes, runny nose. You can also get hives on the skin. We also talked about smooth muscle spasms down here. Okay, so that'll result in things like cramps, diarrhea. You can get vomiting. So you can have symptoms that are mild like this, but you can also have symptoms that are more severe and more fatal. Asthma is one of those symptoms that can sometimes be more severe and more fatal. It's a local inflammatory infection that occurs once you inhale an allergen. So it actually affects a lot of people, 5% of adults and 10% of children in the United States. And basically what happens is they inhale this allergen, and within minutes their bronchioles constrict. So here you see a normal bronchiole, and here you see one that's under an asthmatic attack. And so obviously this type of constriction, you can understand, would cause severe coughing, wheezing, sometimes suffocation. And actually sometimes you can get a secondary reaction 6 to 8 hours after the initial. The problem with asthma is that the treatment regimen often includes more than 8 medications, and so compliance is really difficult for little kids who don't really understand the impact of the medications or don't necessarily aren't necessarily responsible enough to stick with their medications, or low income families that either can't afford the medications or don't have access to the education that they need regarding the benefits of the medications. So asthma is really common or most common in two groups. One of those groups are inner city kids that are exposed to really poor sanitation, really poor ventilation, maybe they don't get enough exercise. And then the other group is the extreme opposite of that. So children that come from extremely clean homes and really don't have the ability to develop that normal immunity. Anaphylactic shock, I don't know if anybody's seen the movie Hitch, but this is a scene from that movie where he is having an allergic reaction to something that he ate, I believe. So anaphylactic shock is a severe, widespread hypersensitivity that occurs really quickly when an allergen is introduced to the bloodstream, and that could either be through the digestive tract or to the bloodstream in some way. So it can result in bronchoconstriction, which we see in this picture down here. Here's a normal airway, and here's a constricted airway under anaphylactic shock, which obviously can result in labored breathing. And then other things that we already talked about, like vasodilation, so maybe you get really red, circulatory shock, and sometimes even sudden death. So we can treat anaphylactic shock with antihistamines, but they're not going to be functional by themselves. Epinephrine is really what helps to subside or combat these symptoms. And then sometimes people even need fluid therapy and respiratory support. So the second type of hypersensitivity occurs when an antibody binds to an antigen. So here's your antibody, here's your antigen, and it activates the complement system that we talked about with innate immunity. So here you get a complement protein binding, which ultimately will result in one of four things that we talked about with innate immunity. So I'll direct you back to that presentation if you want to review the complement system, but that's how a cell dies. And it's just basically an overreaction of this system. So this includes examples like blood transfusions or drug reactions. We'll see this type of hypersensitivity. In the third type of hypersensitivity, you get an antigen-antibody complex. So here's your antigen, here's your antibody, and those will congregate under the skin. Well, this picture is a little confusing because it has complement activation, but basically it can congregate underneath the endothelial cells of either blood vessels or other tissues throughout the body, and then those sites where they congregate will activate intense inflammation, and that can be really damaging to tissues. And then finally, in the final type of hypersensitivity called delayed hypersensitivity, we don't see signs for 12 to 72 hours after exposure. And so this type of sensitivity is what we've already discussed. Antigen-presenting cells display the antigens to a helper T cell, and in response, that helper T cell will secrete cytokines or other chemicals like interferons that'll activate cytotoxic T cells and macrophages. So this is what we see with allergies or to haptens, which are those half antigens that are found or that are not necessarily functional on their own and won't necessarily produce an immune response by themselves, but will when they bind to larger macromolecules. So we find those in things like cosmetics. Penicillin was another example we talked about, poison ivy, and people that get skin grafts and reject their grafts. Okay, so those are all the different types of hypersensitivity. Now we're going to talk about autoimmune disease. So this is when your body fails to recognize your own antigens from foreign ones, and so it'll start to attack your own antigens. So you produce antibodies that attack your own tissues, and that's what you're seeing in this picture here. So here you have normal antibodies that come into contact with foreign antigens. They bind here, and then they destroy the antigens or mark them for destruction and kind of go on about their business. With an autoimmune disease, you have your own antigens coming into contact with your own antibodies. Those antibodies attack your own antigens, and then you get inflammation and tissue damage. So you might be saying to yourself, well, what about negative selection? We talked about how negative selection keeps this from happening, right? It presents your developing T and B cells with cells of your own body, or antigens or antigen-presenting cells of your own body, and if they bind, those cells don't survive. Well, there are 3 reasons that this happens in spite of negative selection. So the first is called cross-reactivity, and that's what you see up here. So with cross-reactivity, you basically have antibodies that react against a foreign antigen, and also similar antigens that are your own. So here we have an antigen that's like violet, let's just say, and this antibody reacts against this violet antigen, and that's a foreign antigen. And then in your own body, you have this purple one. Well, purple is really similar to violet. But this is your own antigen, so it really shouldn't be attacked, but the antibody treats this violet the same as it treats this purple because they're similar, even though this one's a foreign antigen and this one is your own antigen. Another reason that this can happen is when you have abnormal exposure of your own antigens to the blood. So this can happen with sperm. So typically your sperm are kept separate from the bloodstream by a barrier. But if that barrier breaks down and you get exposure of these sperm to the bloodstream, your bloodstream will, or your body will recognize those sperm as foreign and start to produce antibodies against them, which will attack the sperm, and then this can actually cause some men to become sterile. And then finally, we have change in structure of antigens. So all of these little viral viruses here can bind to a host cell, and when they do that, they can actually change the structure of the antigens on this host cell. So now your body won't recognize the antigens that are on this host cell as your own, and cause your immune system to perceive them as foreign. Okay, so that was hypersensitivity and autoimmune disease. So overreaction of the immune system, reaction of the immune system to your own antibodies, and now we're going to talk about immunodeficiency diseases, which is when your immune system isn't responding vigorously enough. The first of the diseases that we're going to talk about with immunodeficiency diseases is something called severe combined immunodeficiency disease, so SCID. And it's characterized by a group of disorders that either result in very little or a complete absence of lymphocytes. So that makes children with this disease highly vulnerable to opportunistic infections, and for that reason they have to live in these protective enclosures. So this is a little boy that we'll talk about in the next slide too, but you can see he's in this protective enclosure, and even when he comes out, he has to live in this little plastic suit. So this disease can be helped with transplants of bone marrow or the fetal thymus, because those are 2 really important lymphatic organs involved in the development of lymphocytes. But they're not always 100% effective, and the reasons for that are that transplanted cells can sometimes fail to survive or to replicate, and also T cells that are transplanted, let's just say in bone marrow from somebody who's healthy, can come into the body and attack the recipient's tissues. So that little boy that we saw, his name is David Vetter. He was a patient that lived with SCID and spent his entire life in plastic enclosures and plastic suits like we saw in the last picture. And he actually died at the age of 12 due to cancer that was triggered by a viral infection that he contracted when they tried to give him a bone marrow transplant from his little sister. So his little sister was carrying this, or from his sister who was carrying this virus, and when they transplanted the bone marrow from the sister to him, he developed a viral infection which resulted in cancer and eventually killed him. Okay, and then the last immunodeficiency disease we'll talk about is AIDS. So, AIDS is acquired immunodeficiency syndrome. It's a non hereditary disease that can be contracted after birth, and it's characterized by a group of conditions that depress the immune system following the infection of the HIV virus. So, somebody starts with the HIV depresses the immune system in ways that we'll talk about as we move through, and then as a result, the person develops AIDS, which is what, uh, well, I don't want to say it eventually kills them. It makes them immunocompromised so that they'll die from things like infection or cancer. All right, so this is the HIV virus, and we're going to talk about here how it invades and replicates in a host cell. So here is the virus, this entire thing, and it basically targets T cells, dendritic cells, and macrophages by binding to them and kind of tricking them into internalizing them. So they're brought into these T cells or other cells by endocytosis. And then once they get in there, they have this viral RNA here, and it's used as a template to synthesize DNA. Okay, so then we get this new DNA that's been produced from this viral RNA, and that DNA is inserted into the host cell's DNA. And once it gets in there, it can stay in there for months to years. Okay, some people live 10 years without ever having developed AIDS from HIV. But when it is activated, it causes the host cell to produce this new viral RNA so that new viruses can emerge from the host cell, which is what you see down here. And then those new viruses are going to adhere to more host cells, repeat the process, and as a result, we get damage to all of the healthy lymphocytes like T cells or other leukocytes or macrophages that are important, and the virus continues to replicate. So the effects of HIV, a person can be HIV positive for over 10 years without ever having developed AIDS, and it's really not understood why that happens, why some people succumb so rapidly and others can be HIV positive for 10 years or so without ever developing the disease. But over time, what happens is the virus, HIV virus, destroys more and more of the healthy cells like T cells. So here in this picture we see this labeled CD4 cell. It's just another name for T helper cell. And so this is a person who has treatment. They have the one virus cell here and a lot of healthy T cells. This is a person without treatment. The virus has replicated and destroyed a lot of those T helper cells, so they have very few healthy lymphocytes. And so as this process happens, people start to get night sweats, fatigue, headache, weight loss, and lymphadenitis, which, if you remember, is just swelling of the lymph nodes in response to battling some kind of antigen. So as these helper T cells that we see here become depleted, the patient obviously becomes more vulnerable to opportunistic infections because it doesn't have as many healthy lymphocytes. So for that reason, death from cancer or infection is inevitable, but it's not really a given when that's going to happen. So sometimes it can occur a few months after the diagnosis of AIDS, and sometimes it happens 8+ years after the diagnosis. So somebody could live 10 years without ever knowing they have HIV or without ever having HIV develop into AIDS, and then people can live a long time with AIDS without ever having succumbed to some sort of secondary infection or cancer. So Kaposi sarcoma is a common cancer among AIDS patients. It's not very common among the general population, but in AIDS patients, you often see it. It originates under endothelial cells and blood vessels, so you end up with these little bruises all over the place and purple lesions they look like bruises. So how is HIV and AIDS transmitted? It's transmitted through blood, semen, vaginal secretions, and breast milk. So what that means is it can be spread from a mother to her baby, either through her placental materials, through childbirth, or through nursing. It can also be spread through contaminated blood products. So since I think probably the 1980s, blood transfusions have been tested for HIV. So now the chances of developing HIV from a blood transfusion are like less than 1%. Drug injections with contaminated needles. So this continues to be the most common way that HIV is transmitted in urban ghettos, is by people sharing dirty needles or used needles to do drugs. It's not transmitted by mosquitoes, so you might be thinking, well, if you can get it from a needle, why can't you get it from a mosquito? If you look over in this nice picture of how you can't get HIV, insect bites is on there. And then finally through sexual intercourse. So worldwide, 75% of HIV infections are acquired through a heterosexual intercourse and predominantly vaginal intercourse as opposed to anal. And so it's not really understood why, but in the US, most cases occur through homosexual intercourse among men. So again, we don't really understand at this point why there are such stark differences in patterns of heterosexual versus homosexual transmission, depending what country you're in. And it's also not transmitted by kissing. So these are other ways that you cannot get it: sweat, hugging, sharing food, toilet seats, bathing, and sneezing or coughing. So the difficulty with treatment and prevention is that 1% of HIV genes mutate every single year, which is really rapid. When an immune cell therefore becomes sensitive to HIV, it's changed within a year. Its surface antigens are different, and so the body can't recognize it anymore and doesn't have any defense against it. So this factor is what also makes it impossible to develop a vaccine. And in this picture over here, we can see the importance of keeping up with medications and drug resistance. Here you can see you have one of the HIV viruses multiplying, and when you introduce a drug, you can keep that replication at bay. But when you stop taking medication down here, now this virus mutates so that it's no longer so that this medication is no longer effective. So now you have two different mutations, and you reintroduce that drug that you stopped taking. Well, that only controls the original form of the virus. And so then you need to introduce a secondary drug, which will hopefully be effective against both of them. So anyways, this is why it's really important to take medications for anything, but especially with HIV where it mutates genes so rapidly. AZT is a drug that was developed, I think, in the 1980s and inhibited that reverse transcriptase process of reproducing the viral RNA. And so it prolonged the life of many HIV-positive people for a really long time, and it was often used in conjunction, sort of like a triple cocktail with two other drugs. But eventually HIV had mutated to the point where it developed resistance to those drugs, and they were failing in over half of patients by the year 1997. Fortunately, today there are 24 plus anti-HIV drugs on the market that are usually used in different combinations. And they are great for reducing morbidity and mortality, so sickness and death, but none of them entirely eradicate the virus. Okay, so practice questions for this section. Why might antibodies begin attacking antigens to which they previously had no response? So if we go back here, you can talk about change in structure of self-antigens, right? Certain viruses and drugs can change the structure of an antigen and cause the immune system to perceive it as foreign. So that's one reason. What is a self-reactive antibody called? I don't know if I put this in here, but it's called an autoantibody. Distinguish between HIV and AIDS. So nobody is infected with AIDS, they're infected with HIV, and then eventually it develops into AIDS. So talk about the difference between the two of them. And then how does AZT slow the progress of AIDS? We talked about this right here. All right, so that's going to be it for today. As always, if you guys have any questions, please email me or we can Zoom or whatever you want to do.